Good day, everyone. I am Joanna Abaday, and together with my groupmates, we will be discussing the backcross breeding technique. This method is one of the most precise approaches used in modern agricultural biotechnology for improving crops and animals. Before we begin, here is our topic outline. We will start with the introduction and history of backcross breeding. Then we will move to the principles and requirements. After that, we will go through the backcrossing procedures, followed by the genetic consequences by repeated backcrossing. And we will also look at the merits and demerits of this technique and present some applications and achievements to show how backcrossing is used in real breeding programs, and finally, we will end with our conclusion. For this discussion, our learning objectives are the following. First, to define and explain the concept and historical origin of backcross breeding in both crops and animals. Second, to discuss the fundamental principles, key parental roles, and essential prerequisites of a successful backcross program. And third, to describe and apply the major uses, merits, and limitations of this method in improving agricultural species. A backcross is defined as a cross between an F1 hybrid and one of its original parents. This technique is widely used in both plants and animals. The main purpose is to retain the valuable traits of the recurrent parent while introducing one important gene or trait from the donor parent, usually a trait like disease resistance, stress tolerance, or a desired physical characteristic. As mentioned, it involves two types of parents. First, the recurrent parent. It is an elite, well-adapted, high-yielding line or breed that we want to keep except it's missing one or two useful traits. Meanwhile, the donor parent is used specifically because it carries the trait we want to add, such as a resistance gene or quality trait. So, in simple terms, backcrossing is like upgrading a high-quality variety or breed without changing its identity, just improving one or two specific weaknesses. Here we see a schematic diagram of the backcross breeding process. First, we cross the donor parent with the recurrent parent to produce the F1 hybrid, which contains 50% genes from each parent. Then, instead of continuing to cross with the donor parent, the F1 is crossed back with the recurrent parent, with its name recurrent, meaning it appears repeatedly. And this is the backcrossing. With each successive backcross generation, the progeny or the offspring becomes genetically more similar to the recurrent parent, about 75% in backcross 1 and approximately 96 to 98% when successively backcross. And this shows how backcrossing restores the genetic identity of the elite parent while retaining the new trait we introduced from the donor parent. Now let's look into the history of the backcross method. Backcrossing is a formal breeding technique was first introduced in 1922 in Washington, D.C. by Harry V. Harlan and Merritt and Poe. They developed and demonstrated this method specifically for cereal crops. Their first notable work was in barley, where they attempted to transfer the smooth-on trait into a popular cultivar called Manchuria. Manchuria already has strong agronomic performance, but it has rough ons, making it less desirable. So by backcrossing the smooth-on donor with Manchuria and repeatedly crossing back to Manchuria, they successfully produce progeny or offspring that look like or similar to Manchuria, having good agronomic traits, but with smooth ons. And this success marked the beginning of backcrossing as a reliable and strategic method in crop improvement, a foundational step that shaped modern plant breeding. On the right side of the slide, you can see the spike and grain structure of barley, and this helps us visualize the trait they work on during those early experiments. Now let's move on to the importance of backcross breeding in both crops and animals. In crop breeding, backcrossing is used to correct one or more deficiencies in elite cultivars. This includes improving disease resistance, seed quality, and stress tolerance. What makes it valuable is that it keeps high yield and adaptability traits intact while introducing the needed improvement. Because of that, it has been widely applied in major crops like rice, maize or corn, wheat, tomato, and many others. In animal breeding, backcrossing is used to introduce or fix specific traits such as milk yield, coat color, or disease resistance. It also plays a major role in breed upgrading, meaning improving local breeds by bringing in genes from superior breeds. These methods is commonly used in cattle, sheep, poultry, and swine breeding programs. Now let's move on to the foundational principles and core requirements in backcross breeding. Again, its primary objective is to improve one or two defects, or we will say weakness of an elite or high-performing variety or breed. And the success of backcrossing depends on choosing the right parents. Here we have the key parental roles. First, the recurrent parent. Again, it is the elite or high-performing variety or breed. It already has most of what we need, but again, it lacks one or two traits. And the goal is to recover almost all of its genetic makeup after the improvement. The donor parent, on the other hand, is chosen because it clearly expresses the target trait, like disease resistance, milk yield, or coat color, and passes it on reliably. And its other characteristics don't really matter or has less importance. It serves mainly as a source of the desired gene. A successful backcross program must meet these four key requirements or prerequisites. First, the recurrent parent must be truly elite, something worth improving. There's no sense backcrossing an average performer, and it needs a lot of trait to be improved. Because again, in backcrossing, it aims to improve one or two weaknesses. Second, the donor parent must express the target trait strongly and ideally resemble or share similarities with the recurrent parent in other ways. This similarity help breeders recover the desired type faster. Third, the trait should have high heritability, controlled by one or a few genes, meaning its traits are passed on from parents to offspring reliably. And this makes the selection easier and the results more predictable. And for the fourth one, or the final requisites, is that breeders usually conduct six or more sufficient backcross generations to ensure that about 99% of the recurrent parent's genome or genetic makeup is recovered, making the improved product or offspring nearly identical to the original or the recurrent parent but more improved. And that covers the foundation of Backcross method on what it is, how it started, and the key requirements for success. And let's move on to the procedural side to be discussed by Daisy.
Good day, everyone. So, I am the one to discuss to you the backcrossing procedures and the genetic consequences in repeated backcrossing. Backcrossing is a practical breeding method used to transfer specific traits from a donor parent to a recurrent parent. The process depends on whether the target gene is dominant or recessive, as this greatly influences the selection strategy, with recessive genes requiring a more complex approach. This means that in plant breeding, the steps we follow in backcrossing are not always the same. If the gene is dominant, mas dali siya i-identify and pilion kung unsa to nga plants ang nag-carry ato nga traits. But if the gene is recessive, the breeders need more generations and careful testing to make sure the trait is successfully transferred. That's why understanding the type of gene is very important before starting the process. Transferring a dominant gene, such as for many types of disease resistance, is the more straightforward application of the Backcross method. Dominant gene transfer is simpler because the trait is immediately visible in the offspring. Since makita na siya sa offspring, so mas dali nato ma-identify si dominant trait. This allows breeders to easily identify resistant plants and continue backcrossing them with the original parent. Since dali raman nato siya ma-identify kay dominant siya, so mas mapadali ato ang pag-select sa mga desirable traits. As you can see in the picture in the right side, this is the illustration of the Backcross procedure that can be used to move leaf rust resistance, the homozygous dominant and the heterozygous from one variety to a susceptible variety, the homozygous recessive. First, the hybridization. Si recurrent parent or si homozygous recessive nga wala siya nag-carry sa resistant trait, and si donor parent, the homozygous dominant nga nag-carry sa resistant genes, imo siyang i-cross, and then the offspring from this cross are called F1 plants or the first filial, since wala dili na ni siya bag sa tua, and this have the combination of heterozygous, meaning they carry the resistance gene, meaning they carry the resistant gene or the heterozygous. Second is the F1 generation. Sa F1 plants or the heterozygous, gi-cross na po siya again to the recurrent parent, which is the homozygous recessive. Again and again, i-cross jud siya sa recurrent parent since backcross procedure or method man ang atong ginagamit diri. So, backcross from the word itself back, so i-cross nato siya pabalik sa recurrent ka parent. Third is the first backcross or the BC1. In this generation, half sa plants will have resistance or the heterozygous, and ang half pod wala siya resistance, si homozygous recessive. The resistant ones are selected si resistance the heterozygous, i-cross na po siya again to the recurrent parent, i-cross siya to the recurrent parent para makontinue ang process. Number 4, the BC2 to BC5 generations. Same rang gyapon nga process ang gina-repeat diri for several generations. Each time only the resistant plants are crossed. Nga generation, ang resistant plants lang ang ginapili and i-cross na pod siya sa recurrent parent. Over time, these new plants become very similar to the original recurrent parent. But karon, naa lang siya na-add nga resistant gene. Next, in the number 5, the BC6 generation, diri nga stage, ang plants are almost the same ra sa original nga variety or the variety A. About 99% of their genes now come from variety A. Gikan siya sa variety A, but they also have the resistant gene from the donor plant. This means they keep the good traits of variety A and gain resistance sa ana nga sakit. Next is the BC6 F2 generation. The resistant plants from BC6, pwede na siya maka-self-pollinate since they are heterozygous or the RR, their offspring will have a mix of plant. Some fully resistant, the homozygous dominant, na apoy uban nga partly resistant, the heterozygous, and ang uban jud dili gyud resistance, the homozygous recessive. The fully resistant plants or the homozygous dominant ang pilion para sa next step na pod nga cross for the next generation. Lastly, the BC6 F3 generation, the dominant homozygous dominant plants from gikan sa BC6 F2 generation are grown. These plants are all resistant to mildew and look just like the original variety A. The best plants are collected and magamit na dayon siya to improve a new variety nga strong and disease resistant since kanang capable na siya ana nga trait sa kapila nga generations nga ila ang pag-cross. The Backcross method can also be used to transfer recessive gene since ganiha ang atong discuss is the dominant transfer of a dominant gene. So now the recessive gene such as one that gives resistance to disease. This process is slightly different because a recessive gene only shows its effect when two copies are present. Duha ka homozygous recessive. First is the hybridization. Dinhi nga part opposite si dominant sa transfer of dominant gene and si recessive gene. This process starts by crossing the recurrent parent or the homozygous dominant which does not have the resistance and the donor parent or the homozygous recessive nga naa pod sa iya ang resistant gene. I-cross nimo siya and this produces F1 seeds. So for the F1 generation, all F1 plants or the heterozygous are susceptible because the resistant gene is hidden by the dominant gene, the R. Katulang isa ka single R nga capital. These plants are then crossed again with the recurrent parent, si dominant, homozygous dominant, para makaproduce na pud sila og BC1 seeds. So for the BC1 generation, the BC1 plants will have a 50-50 ratio of the homozygous dominant and the heterozygous. The plants that are heterozygous gikarry niya ang resistant genes even though dili siya visible. These are selected si heterozygous siya ang pilion na po dayon for the next step nga i-cross siya for the next generation. For the BC1 F2 generation, the heterozygous plants from BC1 gi-self-pollinate siya para makaproduce og BC1 F2 seeds. Ang offspring dayon gikan sa heterozygous plants will show segregation. 1/4 kang homozygous dominant, which is the susceptible, 1/2 pod kang heterozygous, the carriers, and 1/4 kang homozygous recessive, which is the resistant. the homozygous recessive plants are resistant and siya ang pilion for further backcrossing for the next generation na pod. So for the BC2 generation, the resistant plants are then backcrossed again with the recurrent parent, the homozygous dominant. So ang ato ang recurrent parent is si homozygous dominant. I-backcross nato diri si heterozygous para makaproduce na pod siya og BC3 seeds na pod. There is no need to check each plant's genes at this stage. For the BC3 generation, the BC3 plants will again show a 50-50 mix of homozygous dominant and a heterozygous. The process may continue until a stable resistant line is achieved that also looks like the original recurrent parent. Magpadayon gyud gyapon ang pag-cross sa heterozygous or ana nga trait to the recurrent parent. For the BC3 F2 to BC5 F3 generations, in each generation, the plant that show the resistant or the desired traits are identified and backcross with the recurrent parent. Katong na kuha dito nga mga identified nga traits, i-cross na po siya sa recurrent parent. For example, in the BC3 F2 generation, resistant plants are crossed again with the recurrent parent para makaproduce siya og BC4 seeds. Again, i-cross siya sa recurrent parent para makaproduce na pod sa BC4 seeds or the next generation. Kani nga process ginabalik-balik gyud siya from BC4 to BC5 until so on and so forth until the desired trait is stable and most of the genetic makeup resembles that of the recurrent parent. Hangtod nga makita mas successful na nga makita to didto ang katong desirable traits or mga characteristics nga gusto nimo as a breeder. Then next is the self-pollination, the BC5 F2 generation. After several backcrosses sa pila ka generation, plants are self-pollinated para to allow segregation of alleles. This helps identify plants that are homozygous or the homozygous recessive for the recessive gene since the transfer of recessive gene man ang atong gisgutan dari nga part, the desired trait purely. For the testing or the BC5 F3 generation, individual progenies are grown and tested for resistance or the target trait to confirm successful transfer. And this is the last generation nga pag-cross atong mga desirable traits to the recurrent parent. Genetic consequences of repeated backcrossing. So as you can see to the picture in the left side of this slide, naamoy makita nga the non-recurrent parent cross with the recurrent parent. So akong ginayon ganina, katong from the F1, BC1, BC2 generation and so on, ang iyag yung result is the homozygous dominant ni siya nga part. Kada backcross, it reduces the heterozygosity by 50% per generation while increasing homozygosity or the homozygous dominant or the homozygous recessive. Since ang result gyud kada cross nila is the homozygous, it's either the homozygous dominant or the recessive. This means that kada backcross, ang progeny's genome becomes more similar to the recurrent parent, except for the specific recessive gene that's being transferred.
Good day, good afternoon, and good evening. I am Romel Kenth Monceda, and I am going to discuss to you the strategic application of backcrossing. To start, first is the intervarietal transfer. This refers to transferring desirable traits between varieties of the same species. So this also moves simple traits like disease resistance, seed color, or plant height between varieties of the same species. There are types of characters that transferred in intervarietal transfer. First is the simply inherited or the qualitative characteristics, and the other one is the quantitative characters. First, for the simply inherited or the qualitative characters, these are traits that controlled by one or few more genes. For the quantitative characters, this it is measurable, and one example to that is the plant height and also the high yielding. So, also possible for complex traits with high heritability like seed size and shape, complex trait with high heritability. So, complex trait with high heritability. This refers to quantitative characteristics influenced by multiple genes or the polygenic and possibly environmental factors. The high heritability implies a high resemblance between parents and offspring for the trait, between parents and offspring for the trait. Yeah. And let's move on. The idea for the another explanation for the complex high heritability. The high heritability ideal targets is for selection programs because genetic progress can be reliably that made generation after generation despite the traits of polygenic nature. So let's move forward with a beware of linkage drag when unwanted genes come along. So linkage drag is when during breeding some unwanted or harmful genes hitch a ride along with the good gene you want to introduce into a plant. And this happens because genes that are close together on the same chromosomes tend to be inherited together. So even though you select for a good trait, you might also unintentionally bring along some bad traits that lower the quality or the performance of the plant. So next, let's move forward with interspecific gene transfer. So this interspecific gene transfer is a process of moving useful genes like for those for disease resistance from one species to a related but different species. This allows breeders to bring in valuable traits that might not exist in the crop's own gene pool. So the transfer simple traits like disease resistance between related species. So imagine you want to improve a crop by giving it resistance to a disease. Sometimes the resistance gene is not found in the crop species but is present in closely related species. Through interspecific gene transfer, the breeders cross their crop with related species to bring that gene over. To move forward with so these challenges include transferring unwanted genes and variable gene performance in the new species. So this process can be tricky because sometimes unwanted genes from the related species come along as well, so which can affect the crop's quality or growth negatively. Also, the transfer gene may not work as well in the new species because of differences in genetic background. Um, it might express differently or less effectively. Breeders have to carefully select and backcross multiple generations to keep the good gene and remove the bad ones. Next, third one is the improved established varieties. The improved established varieties means adding one or a few improved genes, like disease resistance, to popular crop varieties without changing their main characteristics, or add one or few improved genes, like disease resistance to popular varieties without their overall traits, changing over their overall traits. So this helps maintain their desirable traits while strengthening them against diseases. This approach improves established varieties without changing their identity or key characteristics. So there's an example for the the improved established varieties. For the example of for improved established varieties is the backcrossing two resistance genes, the XA13 or the XA13 and XA21, selecting parent plant the basmati rice as a high-yielding rice. So let's move forward with our fourth one is the near isogenic lines or the NEALs. Near isogenic lines are plants that are almost identical genetically except for one specific gene or small region of the genome. They are created through repeated backcrossing to transfer a particular gene from a donor into an established variety while keeping the rest of the genetic background the same. This makes NEALs very useful for research because um, any differences in traits between the NEAL and the original variety are likely due to that one gene. These help scientists that science studies gene effects precisely and also aids in breeding improved crops with specific traits without altering other characteristics. So to make it more easy to understand, Neals are like twins that differ in only one small detail, like making it easier to study how that detail affects the plant's features. So lastly is the recovered transgressive segregants. These recovered transgressive segregants are plants produced through backcrossing and selection that have traits better than either parent. This happens because the combination of genes from both parents can create new superior traits not seen in the original plants. Backcrossing followed by selection can create plants with their better than both parents. In simple terms, when two different parents are crossed, their offspring can sometimes have traits that go beyond the strength of both parents. By carefully selecting these superior offspring and backcrossing, breeders can develop plants with enhanced qualities like higher yield, better disease resistance, or improved tolerance to stresses. This approach helps create new crop varieties with improved performance, contributing greatly to plant breeding and agricultural progress. So I'll be showing you table 1 example for the achievements. Um, as you can see in the table 1, there is in a column there is crops, the variety evolved traits for BCD, the recurrent parent and the donor parent. So um, the crops for crops there are three examples which are these are the wheat, cotton, and the pearl millet or millet. Then the variety evolved is the new variety that has been developed through backcross. The traits for BCB is uh, on what is the improve from the what traits that has been improved. Like example for the salinity tolerance, the leaf blight resistance. For next is the recurrent parent. The recurrent parent is the variety that has been protected or been um, uh, kept the the background is kept, something like that. And the donor parent is the source of the desired trait. So for the achievements table 1, examples of remarkable achievements through backcross breeding. So crops, first crop is the wheat. So the var so the traits for BCB for the Karsha 65. No, I'll be starting with the variety evolved. So variety evolved Karsha 65. The donor parent is the EG 953. The EG 953 is the traits for BCB. The donor parent is the traits that has been coming from the donor parent is the uh salinity and sodicity stress, and the recurrent parent is the harsha local. Next, in wet crop is the uh INP 852. The donor parent is similar to the first one, is the EG 953, and EG 953, and the recurrent parent is the NP 761. So the trait for BCB that came from the donor parent is the leaf blight and resistance. Lastly, for the wheat crop is the HUW234. The donor parent is PMBWIR4. The donor parent trait is the steam rust resistance, and the recurrent parent is HUW234 as named for the variety evolved. Next crops is the cotton. Uh, to begin with VJ. VJ is the donor parent for VJ is the Goghari A-26 or the Goghari A-26. The donor parent trace is the high gaining altern. The recurrent parent is the brooch DC8 or the BD8. And next to that is the dig VJ. Increased the traits for BCB, the increased fiber length and early maturity from the donor parent of 102 I-1027 A dash LF. Yeah, and the recurrent parent is the V shape. So the third crop is the pearl millet or millet. The donor parent is the MS521, MS541A, and MS570A. The recurrent parent is the TIFT 23A. So the trait BCB of the coming from the donor parents are the downy mildew resistant. And so to continue, I'll be discussing the merits of the strategic application of backcrossing. First of the merits is no extensive field testing needed, since it's an improved version of popular variety and saving 3 to 5 years of uncutting costs significantly. So the second part of the merit is the outcome outcome of a backcross program that can be predicted and it can be reproduced anytime in the future. And third merit is the ideal of using unadapted germplasm, which usually unproductive but valuable gene sources for resistance, quality, or yield traits. And fourth one is it is the only method for the correction of defects. And fifth is the a much smaller population is handled as opposed to the pedigree method. And the sixth is the only method was useful for gene transfer from related species and transfer of the cytoplasm. And lastly, the merits of the strategic application of backcrossing is the modification could be done so that transgressive segregation could occur for quantitative characteristics. And that's it. Thank you so much for listening.
Merits of backcrossing. The new plant variety created through backcrossing is usually not superior to the recurrent parent in overall performance because it only gains the specific trait being transferred, like resistance or tolerance, without improving other characteristics such as yield or quality improvement. And second is along with the desired gene, mga unwanted or harmful nga mga genes that are physically close to it on the chromosome may also be being transferred. So this situation known as linkage drug can negatively affect the new varieties of performance of plants. So third is each backcross generation requires repeated hybridization. Kada backcross nag-require gid siya og hybridization and selection, which is kanang taas kay siya nga process or costly process gid siya. Managing multiple generation and maintaining parent lines demand a lot of time and resources. And lastly, by the time several backcross generations are completed. And lastly, by the time several backcross generations are completed, the recurrent parent used as the main genetic background might already be outdated, or other improved varieties with higher yield or better quality may have been developed, making the resulting backcross variety less competitive. So those are the demerits of backcrossing.
So to conclude, backcrossing remains one of the most precise and reliable breeding technique we use to improve elite crops and animals. Its strength or advantage lies on its focus, in which instead of changing everything, we only correct one or two specific weaknesses, such as disease susceptibility or a performance rate, while keeping all the desirable characteristics of the original line. Through repeated backcrossing, we can almost completely recover the genetic makeup of the, uh, the recurrent parent, ensuring it still performs the same, only better. And even today, with advanced biotechnological tools, backcrossing continues to be essential in modern agricultural uh, programs because of its predictability, efficiency, and proven success in producing stronger, more resilient varieties and breeds. It's a classical method, yes, but still, it is much of a key in agricultural biotechnology.